students, in the last sessions we have studied biotechnology principles and process. So we have studied many process and we have discussed about the principles. But now we are going to study actually what is the biotechnology process. What is DNA and how this DNA or gene of interest it is inserted into the host and how it is produced as a biological product. So we are going to study. First of all, we can understand what are the process of biotechnology. There are different process of bio. So now we are going to study about the process of biotechnology. Process of biotechnology. So what are the different process of biotechnology? You can say isolation of DNA and it is inserting into the plasmid or vector and this plasmid is inserted into the host. So this you can say the process of biotechnology. So first of all, first process we are going to study that we can say isolation of DNA. First process is isolation of DNA. DNA. As we know, DNA, we all very known about the DNA. DNA is the genetic material and it's a blueprint of an organism. It's a polymer of deoxyribonucleic acid. It is made up of polynucleotides. As you know, you have studied already for all the videos, all the chapters. What is polynucleotide? What is nucleotide? Nucleotide contain three components. It is phosphate group, pentose sugar, and nitrogenous bases. So this you can say nucleotide. So here we are studying DNA is a genetic material of almost all organisms and as we know it is present inside the cell. Our body is made up of millions of cells. It is present inside the cell and cell is covered by plasma membrane. In plant cell it is covered by cell wall and inside the cell at the center there is a nucleus. Nucleus is covered by nuclear membrane. Inside the nucleus there is a thread-like structure. It is called chromosome. Inside the chromosome small unit is called DNA. So it is present inside. Actually cell we cannot see by the help of our naked eye. But the DNA is present inside the cell. So from that inside we have to extract out. We have to isolate out. If we isolate this DNA with the pure form, then only it is able to cut by the help of restriction enzyme. So we have to extract this DNA in pure form. How can we extract? How can we isolate? See, we can understand by the help of some steps. First of all, you can understand plant cell is covered by cell wall and fungi cell is covered by the cell wall. It is made up of chitin. So first the plant cell wall is digested by the help of lysozyme. Plant cell it is digested by cellulase. Then bacterial cell wall is digested by the help of lysozyme. Fungal cell is cell wall is digested by the help of chitinase. So by the help of these enzymes we can break the cell wall. Then inside the cell wall, inside cytoplasm, and there are many macromolecules, micromolecules are present. These all we have to digest, and so we have to nuclear membrane we have to extract, and we have to digest, we have to extract out. So when we extract this DNA, along with the DNA, proteins like histone and RNA and other fat or lipids are present. That also we have to remove. RNA we can remove by the help of RNase, and protein we can remove or we can digest by the help of proteinase. So by the help of different enzymes we can digest, we can remove all other macromolecules and micromolecules. At last we will get the DNA, and at last we have to add chilled ethanol. It is called chilled ethanol. So, if we use chilled ethanol, DNA can be precipitated out. Means, you know, when you do the practical DNA extraction practical, at last you add the chilled ethanol. It is used to precipitate. DNA is precipitated out. So, at last we get the DNA. So, this part you can understand isolation of DNA. And the second process you can understand cutting with or making fragments with the restriction endonuclease. We have studied already restriction endonuclease. It is used to cut the DNA with different fragments. So by the help of restriction enzyme, we can cut the DNA. After cutting the fragments of DNA, we have to separate out. That also we have studied in the last videos. By the help of gel electrophoresis, we can separate the DNA fragments. So by the help of DNA means gel electrophoresis, we can separate these DNA fragments by the help of gel electrophoresis. Now we get the DNA fragments that we have to make the copies or amplification of DNA. So next process you can understand amplification of DNA. Amplification of DNA. So amplification of DNA, it means making copies, large number of copies by the help of one of the technologies, one of the tool which is used to make or which is used to amplify the DNA by the help of the technique. It is called PCR. PCR. It means polymerase chain reaction. PCR means polymerase chain reaction. It is developed by one of the scientists. His name is Carey Mullis. Carey Mullis. So PCR is developed by Carey Mullis. By the help of PCR, we will get billions of copies of DNA, our gene of interest. So in the next session, we can study what is the process, explanation of this PCR. Thank you, students.